Okay, in this video we're going to talk about the genetic terminology. If you've been doing the worksheets, you probably kind of have picked up on some of the terminology, but we need to go over it and we need to relate it to physical objects and physical phenomenon, okay, or how things work in the real world. So just because you kind of know how to do it on paper, you also kind of need to know how it works physically inside your body. And when we say something like loci, where is a loci or loci? I really don't know which one is correct there, but what is that? Where is it? What does it mean to be heterozygous? What does that physically mean? So those are the kind of things we're working on in this video. So first of all, we do need to know what a loci or loci is, and that is a location on a chromosome. So the previous video we talked about chromosomes and what they are and everything, and I explained that, hey, there are places on the chromosome that are genes, and they're regular. You can have across different people, the genes will be located pretty much in the same place on a chromosome. So your chromosomes, you have the gene for eye color, all right, or you have the gene for hair color. It's going to be pretty much on the same chromosome in the same spot no matter who you are. And that's kind of important because, after all, when we have children, our chromosomes come together from a male and a female, and basically you have to have your loci, the location, in the same spot or things aren't going to work very well. All right, there are a lot more complicated reasons, but essentially if this got moved down here, there would probably be a problem, and your child would not survive till birth if that happened. So keep that in mind. But basically what we're calling a loci or a loci is the location of your gene on a chromosome. So this might be chromosome 7, and there will be kind of a numbering system for where on the chromosome you are. If nothing else, they'll count the number of DNA nucleotides, all the ACGTs, and they'll kind of, oh, well, this is 7 million from the beginning, and this one is 100 million from the beginning, all right, down here. So they would count it 1 to however long it is, and that is one way to measure where on the chromosome it is, and that's telling you where the loci is, or loci. And that, of course, that means that right in here, you're going to have a data box somewhere here and a terminator in here, somewhere in a little piece of DNA that's wound up, like we saw in the previous video, all wound up in here. And it's hopefully, if it's open, it's running. Like, do we need the eye color gene turned on in a skin cell? No. Do we need it turned on? Do we need the hair color gene turned on in a skin cell? Well, maybe if it's related to producing a pigment that's going to be on the hair, but probably not. I actually don't know in a skin cell. It's possible you need this one turned on, but now that I'm thinking about it, I don't think you do. But that's going to control the pigment that's in your hair. This one's going to control the pigment that's made in your eyes. So this gene will only be, this loci will only be an open chromatin for those cells that produce this pigment right here. And these will only be turned on in the cells that produce the pigment near your hair follicles to produce the color of hair that you have. So keep that in mind, that these might be turned off. These probably are not turned on in the same cells. So these loci in one cell might be wrapped around the DNA very tightly if you're not in the cells that produce this part of your body, but they might be turned on in, or I should say this one might be turned on in certain places wherever you're producing hair. All right, so that's the idea of the loci. It's just the location on the chromosome of your gene. All right, so let's go to the next one here. So here we're getting to these genetic terms that we need to understand. So homologous chromosomes. Well, homologous, it means they have the same loci. All right, and that means they produce the same gene at that same. So you come in pairs, your chromosomes come in pairs, and what makes them homologous is that they have the same genes at the same place. So in the previous video we saw that you have like 23 pair of chromosomes, and all except for the sex chromosomes are going to be pretty much identical. And what makes them identical, it's not just that they're the same size roughly and same kind of shape, but that they make for sure that they have the same genes at pretty much the same spot. So the loci have to line up, and if they do, those are called homologous. It doesn't mean the DNA is identical across here. It just means the DNA can code for a protein, hopefully very similar, but at the same location right here, and usually that is the case. So there'll be a gene here and a gene here, and both of them, one you got from mom, and one, actually this says this one comes from dad, this one comes from mom, and hopefully they're at the same place and they code for the same thing. In this case they code for a recessive gene that they're labeling R. So this is the locus or loci. That's the location, all right, or locations, all right. And here there's the location for the whatever the P gene is, all right. And these two chromosomes are said to be homologous. It doesn't mean they're identical. It just means that the loci for the genes are located in roughly the same spot, and they do roughly the same thing, all right. So now we get on, and so notice here there's a paternal homolog and a maternal. That tells you that I got this one from this chromosome from mom, I got this one from dad. All right, now were they identical? Mostly, but look down here, there's a little c and a big C down here, and so that big C and little c, here's a big C, here's a little c. Well, notice they're not identical. This one is going to have slightly different DNA than this one. The protein will be slightly different, and of course the big C means it is dominant. So we've talked about what it means to be dominant. Basically, whatever characteristic that you get from this, the one for the big C is going to cover up the one from the little c. Now that's a new term we haven't used, characteristic. So the characteristic is like eye color. It's a generic trait. So eye color is a characteristic. The actual trait, all right, here's the hard part. The trait is the actual eye color you have, like brown. So we go back to this one. The trait is brown. The characteristic is eye color. So eye color here is a characteristic. In other words, it's a gene for eye color. That's the generic thing it does. But the specific color you end up with, that's the trait. So you need to know that eye color and hair color are called characteristics. All right, they're kind of like a generic version of the trait. Here you have hazel hair, and here you have brown eyes. Those are specific traits. They may be dominant, they may be recessive, we don't know. You'll have to be told that. All right, so here we go over here, and as you can see, you have two A's, two dominant traits. They're capital letters. So we call that homozygous, which means the same, and dominant. Dominant means big letter. So up here we also have a similar word, homologous. So the word homo means same. So homologous chromosomes mean they're the same chromosome more or less. They're not identical, but they are the same in some kind of characteristic. Or, that's not a good word. Some part of them is the same, and the part that's the same is that they have the genes at the same loci. So here's homozygous recessive. That means they have two recessive genes at the same loci. And then heterozygous means that they have slightly different genes, but still at the same loci. All right, so this one might produce one hair color, and this one produces a different hair color, or eye color, or some other trait: tall, short, fat, skinny, big feet, little feet, whatever we're talking about. And so, but remember, the large letter, at least as far as we know so far. Now we're going to talk about how these two, especially when they're different, can interact. And and that's has to do with whether or not it's completely dominant or just incomplete dominance. So there's complete dominance where this one is so strong it just covers this up completely, so that if this were like, say, brown eyes and this were blue eyes, all you have to do is get this one and you get brown eyes. There's no trace of blue. But what if they were heterozygous and it was not a completely dominant trait? Then you would have an intermediate, say green eyes. So depending on whether or not this eye color, which might be brown and this one might be blue, if you get it like this and it's a dominant, you're going to get brown eyes. There's just brown eyes and then not brown eyes, which is blue. But if they mix, all right, if these two are a mixture, then you might get something else like green eyes. Okay, that's kind of the idea behind incomplete dominance, and we'll be doing worksheets on complete dominance, incomplete dominance, and then we have to worry about sex-linked traits, which are all of these rules, plus you have to add some extra rules if it's on the X and Y chromosome. And then we'll get to some others where we see how traits interact. So that's going to be codominance, and that will be at the end. So codominance is probably the most difficult because you have to learn the others before you can learn codominance. But we will learn all four: complete dominance, incomplete dominance, sex-linked, and codominance. All right, let's move on to another one. Let's kind of enlarge this. Let's see if this can get any bigger. No. Well, all I'm saying here is that when you make a copy of them and they are identical, in other words, they have not just the same loci, which would make it on a homologous chromosome, but you can get a sister chromatid, all right? And a sister is identical. So the sister chromatids are identical, and you make those right before the cell divides, so that the new cell that you're creating has an identical copy of yours. So there is something called a sister chromatid, and it will not be homologous. It will be an identical copy. All right, so just keep in mind that does exist. Not real important, just kind of minor thing that you should be aware that does happen. And here is our idea of homozygous and heterozygous again, just to review it. Of course, you have two chromosomes and you've got big B, big B. Here you've got little b, little b. They're homozygous. So this is homozygous dominant and this is homozygous recessive. So if this, let's say it's dominant, let's say this is brown hair and this is blonde hair. Or let's say this is black hair and this is blonde hair. So this one definitely has black hair and this one definitely has blonde hair because there's no mixing. But if there is such a thing as a heterozygous trait, if you get a mixture of them, well, it might be black hair if it's completely dominant, all right? But it could be that there is a intermediate between the two, and that is called incomplete dominance, and that's when you would have brown hair. Now, I'm going to have to tell you ahead of time, this trait is completely dominant, or I tell you that this trait is incomplete dominance. And if it's incomplete, then this would be brown hair, not black hair. But if it's complete dominance, this would be black hair, just like this one. So in other words, with complete dominance, you just get two what we call phenotypes. We only get two categories of what you can look like. And with complete dominance, you get black hair and you get blonde hair, and here you get black hair again. All right. But with incomplete dominance, this is black hair and this is blonde hair, just like with complete dominance. But over here, you have brown hair, which is kind of an intermediate between blonde and black. So incomplete dominance, you get three phenotypes. Complete dominance, you get two phenotypes. And then there's the other word, genotype. What does genotype mean? It's just the letters. So your genotype is big B, or your genotype is little b, or your genotype is big B, little b. Now the way you say that, of course, is homozygous dominant, all right, or homozygous recessive. This would be heterozygous. So homozygous means the same. So if you have big letters, you have to say dominant. So this person is homozygous dominant. This person is homozygous recessive. And these two people are just heterozygous. You don't say dominant or recessive with this, you just say heterozygous. But here you have to remember to say homozygous dominant or homozygous recessive. We're getting through all this terminology here. All right, so here is an example from, looks like plants, from peas actually. There's a famous guy named Mendel who discovered all these traits, and he studied peas. We'll talk about him. So here are some things that you need to know. You need to know this term allele. And if you have alleles, it just means you don't all have the same genotypes. You have big B and you have little b. So the individual letter like B or T or A, those are alleles. So you have, I should say, big A and little a are alleles. There are different versions of the gene. So you have the A gene, and you have big A, that's an allele, and then little a, that's also an allele. So think allele, think alternate form. It means a different version of the gene, a gene that's typically going to give you a different trait for that character. So your character is hair color, and the trait might be black hair or blonde hair. So this allele, all right, that means big B or little b. And if I'm big B, if I have the big B allele, I will have, say, black hair. Here we see this, you have wrinkled, wrinkled, and round instead of hair color. So if it's completely dominant, of course, you're wrinkled. If it's incomplete dominant, you're going to be somewhere in between wrinkled and round. So these are heterozygous alleles, you know, like big W and little w, or big R and little r. And here they're homozygous allele, maybe that's big R, or big W, whichever letter they want to use you can think of. But these are the homozygous ones, and these are heterozygous. Now, one of them is going to be dominant. I believe wrinkled, I think wrinkled is the dominant trait here. So this one would be wrinkled, and so with this one, if it's completely dominant. If it's incomplete dominant, you'll get something kind of in between the two. All right, so that does it. Those are the terminologies, I should say, the words you need to know for the terminology for the test. So you need to know allele, character, trait, homozygous, heterozygous, dominant, recessive, loci, gene. All of those are things you need. Those are the terms you need to know. Basically, everything we've kind of seen in this, maybe sister chromatid. These are identical. They're not homologous. So there's another term you need, homologous chromosomes. That means they have the loci at the same spot. They have the same gene as far as character goes, but it might be different in terms of trait. So in other words, right here, this little c is for a different trait than the big C. But both of them are for the character that we call, say, eye color. All right, but this would be like blue eyes, and this would be like brown eyes. All right, and that does it for this one.